Hello, everyone. Welcome to today's ESDR Kitchen webinar of the molecular cuisine type. I would like to welcome everyone and remind you that you can ask questions via the chat or the Q&A functions. And without further ado, I would also like to welcome today's speaker, Nicolas Gaudenzio, and head over to Katia as my co-chair, who will introduce Nicolas.
Thank you, Georg. And so it is my pleasure to introduce Nicolas Gaudenzio, who is a research director at the INSERM lab Infinity in Toulouse. So Nicolas, you are also the chief scientific officer at the biotech Genoskin. And so you were trained in immunology, both in France, Toulouse, as well as in Stanford in California. And so your work has nicely contributed to identifying molecular and cellular targets, both in allergic and non-allergic inflammation. And so now, back in Toulouse, you have established your lab and so are developing projects investigating the role of neuroimmune interaction, both in health and disease. So Georg and I would like to thank you for accepting our invitation to share your last results on neuroimmunology and atopic dermatitis. So the stage is yours.
Thank you. Thank you so much, Katia and Georg, for the kind invitation. It's my first participation at the SDR Kitchen. So today I'm going to share with you some data that we had and concept on why do we think that adult and pediatric form of atopic dermatitis or eczema, could be seen as a neuroimmunological disorder. So this is just my disclosure. So in the lab, we study how basically sensory neuron in periphery and immune cells interact all together to regulate inflammation. And we have a strong focus on atopic dermatitis. And so today I'm going to share with you some of our recent findings in the lab and how we think they can make a difference in understanding neuroimmunology in atopic dermatitis. I will cover some concept on mast cell ontogeny and biology because these mast cells are key cells that relay neuronal signal to the immune system and vice versa. And also our recent findings a few months ago about how maternal stress actually can lead to pediatric disorder. We used in the lab mouse models, but also patients' data through collaboration with clinicians, and we use a lot as well the Genoskin ex vivo biostabilized human skin to really understand the translational aspect of our findings in the mice. So I would like first to start by discussing about the peripheral sensory neuron, and just a fresh remember that those neurons, they have their cell body in the dorsal root ganglia close to the spinal cord, and they will basically emit projections, one projection to the spinal cord and the other one basically to the target organ, which will be internal organs, but they can be also the skin. The skin is densely innervated, as we will see later. Those DRGs, when they project to the skin, they can, you can discriminate different type of fibers. We will see that on the next slide. But what is important to understand is that the network of sensory neuron is extremely dense, and it goes all the way up to the epidermal layer. This is how a mouse spine cut open looks like, and you have here the dorsal root ganglia. So it really looks like a small lymph nodes with a lot of diversity of sensory neurons. And now, based on single-cell RNA-seq data sets, we know that there are different subtypes of sensory neuron: threshold mechanoreceptor, peptidergic neurons, non-peptidergic neurons, etc. We can discriminate them based on innervation territory, transcription factors, surface receptors, neuropeptides they secrete, etc. And today we will cover, for example, the threshold mechanoreceptor, non-peptidergic, but also the PEP1 neuron that is a producer of substance P. So talking about substance P, in mast cell, which are exclusively tissue-resident immune cell and highly enriched in the skin, there is the expression of this receptor named MRGPRX2 in human or B2 in the mouse. Its discovery by the group of Qing Dong in Johns Hopkins University has really broadened our view of mast cell biology and how they actually regulate skin homeostasis. And this receptor, MRGPRX2 or B2 in the mouse, is the mast cell receptor for substance P. For example, mast cells do not express the neurokinin 1 receptor, which is expressed by other cells. And MRGPRX2 is B2 in the mouse is exclusively restricted to mast cell. In human, you can find it as well a little bit on activated basophils and somehow on eosinophils, although their function is not known, but we do not find it in human sensory neuron, for example. Then when you engage this receptor with, for example, substance P, but it can bind many additional cationic molecules, then you trigger a massive degranulation. And this is how a primary human mast cell look like if you incubate it with substance P. You will see that they will flux the calcium and there will be a massive release of granule-associated mediators containing histamine, proteases, inflammatory substances, etc. In 2019, using a model that mimics adult atopic dermatitis in a way that you have an epicutaneous sensitization of the skin with house's mite, staph aureus toxins, and so you develop an antigen-specific atopic dermatitis. What we found is that mast cells that express MRGPRX2 or B2 here because it's a mouse mast cell, they form what we name sensory cluster with peripheral neurons. And those sensory cluster, we found that they are actually capable of directly detecting allergens thanks to the sensing of the proteases from allergens. And these allergens will activate sensory neuron secretion of substance P that will then trigger the activation of the nearby mast cell via MRGPRB2. And this degranulation and this spark of inflammation was actually completely required to trigger the development of atopic dermatitis-like pathologies. If you don't have this interaction, then there will be no further development of type 2 immune response, for example. So we also did some intravital imaging, and we showed that if you engage sensory neuron, you will find that the mast cell nearby start to degranulate. So based on these findings, we proposed that MRGPRX2 was a new therapeutic target to treat mast cell-dependent inflammatory skin disorders. We collaborated with some biotechs in the US and to help validating some small molecules. And now there are 6 ongoing phase 2 trials in AD and chronic spontaneous urticaria. We're very much looking forward to the results. So we were not the only one to publish this in 2019. 3 other papers by our collaborators from Johns Hopkins University, Xing Dong, or Brian Kim from the Mount Sinai. This one is in collaboration with our lab as well. Show that this mast cell neuron coupling is actually key to regulate many aspects of skin homeostasis and pathogenesis. For example, pain sensation, but also to drive non-histaminergic itch, and also to help to fight against bacteria. So we wanted to understand a little bit more also how all of this would be happening, and for that we had to take a step back and understand, the ontogeny of those mast cells, where they are coming from, why can they interact with those neurons. So this is the work by Lilian in the lab and Marie, who is now in Lyon and has our lab and project. The idea was to really decode the ontogeny and the transcriptomic heterogeneity in mast cell in mice and humans. So just here, it's going to be my only slide on this project, but it's going to be important for the rest of the talk. Those mast cells that express MRGPRB2 and that we can classify them as neuron-associated mast cell. They are the only one that express MRGPRB2 that can respond to substance P. They have their own transcriptomic profile. They are really long-lived. You will find them in the tissue for months before being replaced. They will seed most of the tissue, and in all the tissue that you will look at, they will be always in contact with neurons. On the other side, you have mast cells that do not express MRGPRB2. Textbooks were saying that they were coming from, basophil mast cell progenitors coming from GMPs. We actually use different fate mapper system. We found that they are not coming from GMPs, so we are still trying to identify where they are coming from in the bone marrow ontogeny system. We know that they develop after birth or shortly at birth. You need microbiota-derived signal, and they will seed mostly the gut and a little bit the lungs as well. However, during inflammation, and that's the work by Kenji Kabashima in Japan, that you have a recruitment of bone marrow progenitors that will then adapt the same phenotype and adopt the same phenotype, that B2 positive mast cell, and they will be able to respond to substance P. And that was in a model of atopic dermatitis. So having this in mind, I would like now to switch to our recent findings about, why do we think that basically pediatric atopic dermatitis or eczema, we think we should not call it atopic dermatitis for this very specific model, is actually a neuroimmune disorder, and it starts actually before birth. So it's the work by Nadine Serrin, who is a postdoc now at Myriam Merad's lab in New York, and it's a huge team effort, obviously. So, as you know, I'm not going to introduce atopic dermatitis to this audience, but 20% of infants develop what we name signs of eczema, in the first 4 years of life, and for some of them very quickly, like just a few days after birth. It's characterized by tissue inflammation, increased mechanical sensory sensitivity, meaning that the sensory system might be also overactivated, which can worsen, itch, disrupt sleep, and impact the quality of life. And if we look at this nice graph about, the onset of atopic dermatitis based on the age, and we are very interested by this very early onset of pediatric eczema with remission. So for 70% of the kids around the age of 4 to 7, basically most of them, they will go in full remission of the disease. So we wanted to understand what's going on, and there were some epidemiological studies that show that basically maternal exposure to stress could increase the risk of eczema in kids. So we wonder whether environmental perturbation in utero could predispose to pediatric eczema at birth. For that, we developed a model in collaboration with Nicolas Senac. He's now part of our institute at Infinity, in which we expose pregnant lambs, 3 times per day for 30 minutes for 6 consecutive days to bright light, and then we analyze what's going on after birth in the offspring. So what we found is that if you have a PEP that is coming from a controlled pregnancy or a stressed pregnancy, there is kind of the same. The skin looks kind of the same, no signs of inflammation, and you have a normal percentage of immune cells. However, something that we did not take in consideration at the beginning is that pediatric eczema occurs usually at flexural location of the skin, often exposed to constant mechanical friction, such as also the wet environment of the diaper, for example. So we wanted to mimic this, and what we did is that we just patched PBS filled gauze pad occluded with a dressing, to mimic a light, wet mechanical friction. And if you do that on a mouse that is coming from a controlled pregnancy, nothing happens. The skin is totally normal. However, if you do that on a mouse that is coming from a stressed mother, you will see that it's going to start to develop a very severe inflammation. And what is even more fascinating is that exactly as it's seen in infants in the clinics, if you let the mouse grow older, then the phenotype naturally reverses with the age as well. So we said, okay, we might have actually a model that mimic this kind of, hyperreactive skin at birth with remission with the age. And we wanted to dig a little bit more into the mechanism of action. So, the way that our body sends, mechanical information is through sensory neurons. So what we did first is that we simply stain for sensory fiber projections in the skin tissue. This is if the offspring is coming from a control mother. In green, you have the beta tubulin that stains for most of sensory neurons. In red, you have tyrosine hydroxylase that will stain for celothreshold mechanoreceptor, but also from sympathetic fibers in the skin. And if you have an offspring coming from a stressed mother, we found that there is a completely enhanced branching and density of all the sensory fibers within the skin tissue. We also found, not going to go into detail here for reason of time, but we also found that it's associated with differentially expressed genes and transcriptional reprogramming of those neurons. enhancement of the activation of the neuron responsible for conveying sensory mechanical information. On the inflammatory part, we found as well, we perform a single cell RNA-seq on CD45 positive cells isolated from the skin tissue. We also found that there is a normal distribution of cells, like by flow cytometry. However, when we analyze the genes of those cells, we found that actually mast cells were profoundly impacted by the prenatal stress. So all the mast cells in the skin of those pups after birth are completely dysregulated. When we look at the genes, these are genes associated with granule exocytosis, activation, energy metabolism. And when you look at the face of mast cell as compared to a control skin, if the pups are coming from a stressed mother, mast cells are already degranulated at steady state in the absence of stimulation. So do we think that actually those mast cells are responsible for a phenotype? So to answer this question, we use two complementary models of mast cell deficient animals. We use the KIT WSH mice, in which, there is a profound lack of mast cell, but also they have a splenomegaly, more basophils, etc. So it's not a perfect model, but we found that those mice were fully protected from the development of the pathology. Then we use the MCPD5 Cree DTA mice from Axel Rohr's lab in Germany. MCPD5 is a protease that is selectively expressed by those mast cells that are of primitive hematopoiesis and that express MRGPA-B2 and that are associated with neurons. And when we selectively deplete those mast cells, the mice were also fully protected from the development of the pathology. So you need those mast cells to develop the disease. So what is going on? So to better understand what's going on, we actually took a step back and we look at really right after the stress event in the mother and in the fetuses, what is going on. So this is a cross section of an embryo, coming from a control mother on the left side, coming from a prenatal stress mother on the right side. On the left side, you can see that those mast cells that feed the skin, mast cells are coming first of all from the yolk sac, then from the HACs a little bit later during the development, and they seed very quickly the skin like macrophages in the same time. And if they are coming from a non-stressed mother, they are not degranulated. If they are coming from a stressed mother, basically those fetal mast cells are already degranulated at steady state. And these are the ones that will actually persist after birth for a little bit of time. So we sorted those fetal mast cells. In utero, they do not express the high affinity Ig receptors, so you have to sort them based on the stem cell factor receptor and IL-33. And then we perform a bulk RNA-seq analysis, and we found that indeed there is more than 1,000 genes that are differentially expressed in the fetal mast cell just by stressing the mother. These were genes, again, associated with granule formation, remodeling, maturation, but also, activation of the cells. So what is the molecular mechanism that actually regulates this activation of mast cell? So, in the body, we have two different types of stress response system. You have the HPA axis that leads to corticosteroid single synthesis and that acts through the glucocorticoid receptor. And on the other side, you have the secretion of noradrenaline and that acts through the adrenergic beta receptor 2. So we then took advantage of those single cell data set. Do you see my screen? Okay, single cell data set of the developing skin. We isolated mast cell based on the carboxypeptidase A3 expression and as a control also macrophages based on CSF1R. This is a control of a population that developed exactly in the same time. So what we found is that mast cell, they do express very high level of the glucocorticoid receptor and L3C1. However, they do not express the adrenergic beta receptor 2, so only macrophages actually express it. So to verify whether this HP axis was activated when we stress the mother, we dose corticosteroid in the amniotic fluid of the pregnant dams, and we found that indeed by stressing the mother with light, you have a huge increase in corticosteroid in the amniotic fluid of pregnant animals, and those babies, the embryos, they basically bath in the amniotic fluid, they even swallow it, and the skin is exposed, to this really corticosteroid enhanced amniotic fluid. So to really understand whether those fetal mast cells could react to glucocorticoid, We sorted fetal skin mast cell, incubated them with, either vehicle control or corticosterone. They do degranulate and respond to corticosterone. If you incubate fetal mast cell with the amniotic fluid from a control mother, nothing happens. If you incubate fetal mast cell with the amniotic fluid coming from a stressed mother, they start to degranulate. So how can we interfere with that? So we use the metirapone. Metirapone is an enzyme, a molecule that selectively inhibits the enzyme responsible for corticosterone synthesis. We use a dose to be injected just after each stress event in the mother that would actually not completely block but normalize the peripheral level of the corticosterone in pregnant animals. Here you have the peripheral blood level of corticosterone in control mother that are stressed in red. And then you can see that when we pre-treat with metirapone, we actually go back to kind of a basal level of a non-stressed animals. So when we did that, the effects were dramatic. We completely blocked the development of pediatric eczema, no more increase in epidermal thickness upon light mechanical friction. You restore a normal mechanical sensory information in sensory neurons. When you look at the face of mast cell, you also completely restore a normal phenotype. They are not hyperactivated and degranulated anymore. So to investigate the translational aspect of these findings, what we did is that we took advantage of this nice single-cell data set of human fetal skin development and immune cell in particular. And what we found first is that exactly like in the mouse, mast cell and not macrophages are the only one that express the glucocorticoid receptor, while macrophages can express the adrenergic beta receptor too. And atopy and stress is a long story. We know that atopy is associated with heightened anxiety and stress-induced exacerbation of condition, as well as increased risk of eczema in offspring. We also know from the literature that newborns with a family history of atopy have an enhanced HPA axis response as well. So we started a collaboration with Jared Chan, Anand Andyapan, and Florent Ginu with this cohort named Healthy Early Life Moments in Singapore, in which they nicely follow actually couples before conception, then during pregnancy, and then they follow the kids after birth now. So we had access to blood sample from pregnant women at the gestational time or during delivery, because we know that at delivery there is a huge boost of cortisol, actually natural boost. And then those women could be, classified as atopic women or non-atopic based on the positive, basophil activation test against house dust mite and global level of IgE against house dust mites. And what we found is that atopic women during the gestational time and during the development of the immune system, again, they have more cortisol than non-atopic women. However, when you look at during the delivery time, then there is such a huge secretion of cortisol by the body that you cannot spot any difference anymore. So based on these findings, we wanted to propose a revisited version of the etiology of very early pediatric eczema with remission that would be centered more around in utero dysregulation of neuroimmune circuits. In other words, you have psychological stress that will lead to HPA axis in the mother, then influx of corticosterone or cortisol in human in the amniotic fluid, and then this will completely reprogram yolk sac-derived mast cells. These I didn't show you, but these are actually the yolk sac-derived ones that are sensitive to corticosterone. But also, sensory neuron responsible for conveying mechanical sensory information. Then you will have a kind of a hyperactive skin at birth to mechanical stimuli. And what I also did not show you was that basically those fetal mast cells that are dysregulated, they persist after birth, but then after 6 months in the mouse or several years in humans, they're actually replaced by new mast cells that are not exposed to endogenous stress anymore. These are normal mast cells, and then you go in full remission of the pathology. And with that, I would like to thank the people in the lab who perform most of this beautiful work. So Lilian, Nasser, Marie, and Nadine, and also all the people that participated to the lab, our precious collaborators. our funding agency that are generous when they want, and all the people at Genoskin for the translational samples. And postdocs are wanted, so feel free to apply. And thank you very much for your attention.
Thank you so much, Nicolas, for this really inspiring presentation and all the data you showed and mechanistic studies.
Thank you.
I don't see any questions yet, so maybe I can start with one question. So the stress in neonatals is how do you think that this really modulates the immune response on a long run with a chronic that leads then to a chronic disease like atopic dermatitis? So do you think that this is also mast cell driven, that they are kind in the more hypersensitivity state or so? Or do you think that mast cells modulate other immune cells or maybe structural cells to then become more, the risk is higher than to develop such a chronic disease?
It's a very good question. So what we show already in the paper is that this corticoid imprinting triggers an epigenetic remodeling of mast cell, and they will stay as dysregulated before being replaced, basically for several months in the mice, and we think this is a year scale in human. The most important is that this is not necessarily the driver that will be then switch the pathology in which in a chronic disorder, antigen specificity, and etc. But we think this hyperactive skin is actually leaky. If you look at the transepidermal water loss, it's going to be very increased, and tight junctions, etc. So we think it gives a window for the kids at birth in which they get sensitized, and it turns the disease into then a chronic disorder, what we know of atopic dermatitis, basically. We think this is one of the reasons why, and these are still ongoing projects in the lab, but one of the reasons why you can switch, the first manifestation would be the skin, and then after, basically, you will switch into the atopic march type of disorders. Yeah.
Very good. So there are a few questions now in the chat, actually. So one is from Johanna Strobel. How would you hypothesize that iatrogenic corticosteroid treatment during human pregnancy actually influences mast cells?
Yeah, it's a very good question. So we are currently discussing that with our colleagues in Singapore and in the cohort, actually. So if you have treatment with corticoids just before birth, for helping maturation, etc., I think in the window it's too late to deprogram mast cell because they're already there, and these are not yolk sac development anymore. There is a susceptibility window. However, what we are more concerned about is the treatment with dermocorticoid during the pregnancy time, of the mother. And we are trying now to look whether there would be an eventual relationship between dermocorticoid treatment and the development of pediatric form of early eczema at birth. But this is still ongoing, and I don't have the answer yet.
Another question, as I understand, mast cell numbers are increased in general in atopic dermatitis skin. Are these recruited from the bloodstream or proliferating locally in skin? What is the role of mast cells in adult atopic dermatitis?
There is an increase in mast cell in atopic dermatitis, also in urticaria, for example, in CSU. Those mast cells, they are actually coming. At least from our mouse studies, but they are coming from the bone marrow. So you have the local resident mast cell that are kind of self-renew and very long-lived, but then you have a recruitment of bone marrow-derived progenitors that when they reach the skin, they adopt the phenotype of skin mast cell, and after they are not dissociable from the previous one. We know that mast cell play a role, in atopic dermatitis. So we think it would be through the inflammatory symptom, but also the sensory and itch symptom. They are able to drive, also histamine independent itch via the secretion of proteases, and notably via the engagement of MRGPRX2. And this is one of our work that we published with the company Science Pharmaceutical in JACI a few years ago, showing that if you have a humanized model or a mouse model, basically of atopic dermatitis, if you use a small molecule that blocks MRGPRX2, then you might be able actually to block either the development or the chronicity of the pathology. And now there are ongoing clinical trials. So the true answer in human will come, I would say, next year. Yeah.
Okay.
We do have a question also from Denis Knikin asking on which day mast cells populate the fetal mouse skin.
So so it also depends on which fate mapping system you're using, because you can get differences. But usually there is a consensus that the first one are yolk-sac-derived mast cells, at least the first wave, and they arrive between E7.5 to E8.5, like macrophages.
So Maria Bodnari is asking, how does the cortisol penetrate the skin of fetus to access the mast cells?
Very good question. I don't have the answer. Uh, I don't have the answer at all, actually. So we did not study really. What we know is that if you take those fetal pups and you dose the cortisol, it's really elevated. So somehow it penetrates. I don't know how it is possible. Maybe I don't know if they swallow or if this is a direct contact through the skin, and the skin, is not like a super thick. At least in the embryo, it's very small and smooth. So maybe through this mechanism, but I'm not sure it's an active mechanism.
Also, Sophie Hu is asking, based on your results, if you could comment on the corticoid treatment on atopic dermatitis children.
So I think after birth, it's okay. We think that also if you take mast cell from, at least you derive it from peripheral blood, human peripheral blood, so after birth, those mast cells, they do not degranulate in response to glucocorticoid. And on the contrary, it's a little bit more anti-degranulation. So we think that actually the glucocorticoid, and that's an ongoing work in the lab, it's that glucocorticoid imprinting by the mother is part of the education of the yolk sac progenitor from the fetus. Actually, it's kind of the first education that gives the mother to the immune system. And it's only those primitive progenitors that would be susceptible to glucocorticoids. So I would not necessarily be afraid of treating infants with glucocorticoid.
To stay on time, it will be the last question from Alain Tayeb, who is asking what is known in humans on the equivalent of those yolk sac mast cell situation in the dermal compartments of newborns and link with neurons.
Yeah, so it's a good question as well. So when we switch to human and ontogeny, it's a little bit more complex, obviously, because we don't have fate mapping system. However, based on fetal tissue analysis, we know that you have the exact same yolk sac-derived mast cell that will develop very early as well. In human, and they develop, in the mouse, they just have like 5-6 weeks before birth, or 5-6 days, sorry, before birth. So they don't stay in the fetus for a long time. I mean, per se before birth. However, in human, it's more than 3-4 months. So then they have the time to be dysregulated. We still don't know in human whether those mast cells are the one that stays that populate the skin at birth, because we don't have fate mapping system. So this is something that we still need to investigate. But the important thing with the sensory system is that mast cell and sensory neuron, they develop in the same time in mouse and in humans in the skin. So neurons will be starting to branch and mast cell will be starting to develop. So we think those cognate interactions actually are happening in utero already before birth. But in human, it's a little bit more complicated, obviously, to analyze.
Okay. Thank you very much, Nicolas. And so everyone is congratulating you and really enjoyed your presentation. So.
Thank you.
Yeah, thanks a lot.
Thank you very much.
I'd also like to thank everyone who was involved with the many questions that were asked. So this shows how interesting this topic is and how much we can still learn and discover in this field. So I would like to remind everyone that the webinar will be open access as a webcast in a few days. And then also already announced the next ESDR Kitchen webinar, which will be of the freshly baked type. So a very recent publication will be presented and discussed on the 11th of March. And then another reminder today is the Women and Girls in Science Day from UNESCO and UN. And also as ESDR, we are fully committed to this topic. And there is a nice campaign that was launched on the ESDR homepage. And I would like to invite everyone to go on the homepage, look at it, and eventually then also massively reshare some of the ideas then with social media so that we make some noise in this regard. And then finally, I would also like to mention that the abstract submission and registration for the ESDR annual meeting is open since yesterday. And I hope to see many of you in person in September in Heidelberg. Thank you.